What gene therapies can do is it can change those errors, sort of like taking an eraser, correcting those spellings, and then your meaning of the sentence is restored. That's what gene therapies can do. It can bring back those normal functions.
Welcome to Science with a Twist, a podcast for curious people who enjoy exploring how science impacts our daily lives. From innovations in biopharma and diagnostics to technology that improves food safety and fuels advances in clean energy, it's all thanks to science. In each episode, members of Thermo Fisher Scientific's team talk to experts who are defining the next advances in science and technology that enable us to thrive. Let's get started.
Welcome to Science with a Twist. I'm your host, Terry Summers. Today, we're diving into the revolutionary world of gene therapy with our guest, Namritha Ravinder, Director of Cell Biology at Thermo Fisher Scientific. Imagine a future where diseases that were once considered incurable are now treatable, where personalized medicine can target and eradicate illnesses at their genetic roots, and where groundbreaking technologies like gene editing are changing the face of healthcare. These aren't just dreams, they're happening right now. Join us as we explore why the buzz about gene therapy is more than just hype, and how scientific advancements in this area are set to transform lives and redefine medicine. Our guest today, Namritha Ravinder, PhD, is the Director of Research and Development at Thermo Fisher. In her current role, she oversees a product development team that enables closed, automated manufacturing of cell and gene therapies. Initiatives like these are at the forefront of transforming complex, labor-intensive processes into scalable, efficient solutions that can help bring the promise of cell and gene therapy closer to reality. Welcome, Namritha.
Thank you, Terry, for that nice introduction.
I'm looking forward to this conversation, Namritha. It's such an exciting time for this promising field, but it's complex science. And a non-scientist like me would love and appreciate your explanation of this. So let's start with the basics, if we can. Would you explain what gene therapy is?
Sure. Imagine a world where diseases like leukemia, sickle cell anemia, and multiple sclerosis, which have debilitating symptoms, are no longer life sentences, where we are not just treating those symptoms, but rewriting those underlying informations or instructions that are changed and then manifested into these diseases. And you can restore this health permanently. You know, a child that has symptoms for lifelong and has to go to multiple hospital visits to treat sickle cell anemia symptoms, if you can give that child one treatment and cure that disease, wouldn't you want that therapy? And that's the kind of promise, a new hope to life that we're talking about. The way this works is, just to simplify, our body is made up of building blocks called cells. And these cells have large instruction manuals called DNA. And these instruction manuals provide all the information needed for the body to grow and function and do what it's doing. And these informations are separated in small, tiny chunks called genes. And occasionally what happens is inside these genes, there are some errors. Think of it like spelling errors that change the meaning of the sentence and the function of the cell, which then becomes the disease itself. What gene therapies can do is it can change those errors, sort of like taking an eraser, correcting those spellings, and then your meaning of the sentence is restored. That's what gene therapies can do. It can bring back those normal functions. And another thing that it can do is you can add new functionalities into the cell. Think of an immune cell that in your immune system that can naturally go fight foreign infections. What you could do is you could leverage those cells, provide them new functions. Sort of like you're training your army, and then now this army is trained to go fight. You can actually now engineer these cells with gene therapies, provide new functions, and now these immune cells, when it's back in the body, it can go hunt for those cancer cells and clear them. That's the promise of these gene therapies.
So these genetic fixes, are they occurring inside the body or outside the body?
Well, there are different ways the researchers can handle this, and there are different technologies. Now, just to simplify, there are two types, right? In one process, what you could do is you can take the cells, the building blocks that have this instruction manual outside of the human body. In that case, you would call it ex vivo gene therapy or ex vivo cell therapy because you're taking the cells out and tinkering the genes. And what happens in that scenario is, someone in the lab can take those cells, provide those cells with this new genes. Maybe it is a disease that has this genetic mutations or errors in the instruction manuals that you're correcting, or sometimes you're just giving new functions to the cell. You can actually arm your natural immune system and make them these trained armies that can go hunt the cancer and clear those cancers. So all of those things can be done outside of the body where you're processing the cell and then give it back. You could also do it in vivo within the body. In that example, what one would do is you directly provide those or deliver those genes with the new function or a corrected gene directly into the tissue or the organ that you want to treat those diseases in.
How long has this science been around?
The science has been for a long time. I mean, if you go back to the early researches in 1960s, where researchers tried to test this out in the lab, taking mammalian cells, meaning human cells, outside of the body, testing them, and then see what things can be done with the genes. But really, the breakthrough came in around early 1990s when William Anderson and his team secured clinical trial approvals for a disease called SCID. And here, this disease is commonly known as a bubble boy disease. What happens is these children, they have compromised immune system, right? Even a simple cold can be life-threatening for them. So they're staying in an environment which is completely sterile, like a bubble. Hence the name of the disease. So what researchers have done for this particular clinical trial was, it's really cool. They are actually taking those immune cells, providing those genes that cause those disease symptoms, and then correcting them, and then putting those back into those patients. And now those symptoms are permanently cured because you have corrected the underlying cause of that disease. That was one of the earlier clinical trials, but the field has moved so far in these last 35 years that you're seeing more breakthroughs where one can treat cancer by reprogramming or engineering the cells of the immune system to go hunt for the cancer tumor and then clear it. Treat some common diseases like maybe autoimmune diseases and others. And really, the science has advanced there quite a bit.
And we talked in the introduction about how this is an area of research and medicine that's really at the cusp of transformation. So how many gene therapies are on the market right now, and what does the pipeline look like?
There are a number of applications and number of clinical therapies out there, over 30 approved therapies. And I think the latest data that I've seen from this first quarter of this year is 33 globally approved gene therapies. And then if you look at how many are there in the pipeline, there are over 2,000 gene therapies that are in development, either preclinical or clinical trials, quite a bit. And this spans liquid tumors, solid tumors, some emerging ones for beyond the cancer diseases as well.
Yeah. Can you give us some examples of some of the early gene therapy successes?
I'll talk about recent, in the last decade, approved therapies. One of them is Kymriah from Novartis. It's an approved ex vivo cell therapy, where you're taking the lymphocytes, the T cells outside of the body, giving them new functions. Sort of these trained armies that I said can go back into the cell and then go hunt those cancers and clear. That's the example here. And this is for ALL, leukemia, blood cancer. And then another example is Luxturna. And this is from Spark Therapeutics. Here it involves in vivo therapy. So you're actually taking the gene and delivering it to the retinal cells, the cells inside the eye. And then you are treating for an inherited genetic disorder that has progressive vision loss. So once you provide these genes in those retinal cells in the eye, then you can restore the normal functions, and now you have that cure. So those are some examples of therapies that have been approved.
So you're talking about making changes, these genetic changes, at the nanoscale level, right? So what kind of tools are you using to make these very precise changes at such a small scale? How does that work?
There's a lot of ways you could do it. One of the recent ones, the technologies that really boomed and got this gene therapy, cell therapy era into a new phase is CRISPR technology. So it's a gene editing tool, as the name suggests. It can edit the genes within those instruction manuals to restore the functions or provide new functions. So what this does is think of it like a eraser, right? You put that in the cell or the body. It goes into the cell where it needs to using the natural mechanisms and then finds that instruction manual, scans through the DNA, the instruction manual, and now finds that precise gene that you want to edit, either to correct a gene mutation or add a new function. And once it does that cutting, and then the rest is magic that happens within the cell. If you provide, along with the CRISPR, a corrected gene or a gene with new function, after CRISPR cuts the gene at the required site, this new function can be added or new change can be added. It's interesting because how these technologies are not what was normally found in humans, right? This was originally found in bacteria.
Ah,
And then
okay.
researchers found this cool function and they said, okay, now why don't we try this for these editing genes within human cells? And this is really groundbreaking. In 2015, Science Magazine named CRISPR as a breakthrough technology of the year, opening new doors for new therapies. There's, a cool one that was recently approved, and this was in the news, is Enchelto from Neurotech. This is cool because I found it interesting because these are therapies that you can encapsulate in tiny capsules, which are then, inserted into the eye. And then you can, there is the magic happens there. Really cool science.
It sounds cool, except for the inserting into the eye part. It does it. I'm not comfortable with that, but if it works, yeah, it's worth it. That's really fascinating. So, Namritha, do you do work related to CRISPR, and is Thermo Fisher involved in CRISPR technology?
Yeah, Thermo Fisher is involved. Outside in the field, there are a lot of inventors of these technology, CRISPR-based technologies of different kinds. At Thermo Fisher, we leverage these technologies and build products. So we have several products that can enable our customers to do gene editing. And it's all the way from research use grade reagents to the clinical manufacturing scale reagents that are compatible for both ends. And then your other question about did I have the opportunity to work? Yes, I did have the opportunity to work on CRISPR-based products. And one of the first products that we had from Thermo Fisher, I had the opportunity to develop that particular product that we had. And then now in my current role, my team and I, we are leveraging CRISPR-based products to build instruments to test out these technologies that will help build cell therapy manufacturing and gene therapy manufacturing. So we get to use these technologies on day-to-day basis.
I imagine Thermo Fisher has these tools, and these tools that ensure the DNA changes are incorporated, correct, and performing as intended.
Several tools. And if you think of this whole process that I was mentioning, sounds very cool, sounds very interesting, but it's not that easy, right? There are multiple different steps along the way. You are isolating the cells if you're doing ex vivo or injecting the gene, but you need that gene that will bring that new function. So those genes are synthesized in labs, and then you need tools to make sure that you have the right spelling, so that gives you the right function and the right meaning. So technologies at Thermo Fisher, we have sequencers like S5 sequencer, which can sequence the DNA, sequence the gene before it's introduced into the cell. And that's one thing, right? And then once the gene enters, you have to make sure that it is actually doing what it's supposed to do. It gives the right function. It's expressing the right protein that it's supposed to. The gene has to translate into a protein that will then do whatever magic it needs to do in the cell. So there are technologies where you can look for these proteins, right? Some of these, like you mentioned earlier, the radars on the immune cells, you need to check this radar is present on the cell or not. So there are technologies like flow cytometry. So one example at Thermo Fisher, we have Attune CytPix. And these are instruments that can look for these protein markers on the cell and then say, okay, these cells are positive for these markers and it's showing those functions. and several assays that you can run in the lab, experiments to see if the gene that I had put into the cell has expressed, so Tachman assays and several of those. And the other thing is, these cells, before you put them back, you are expanding them to make sure that you have enough dosage of these cells to treat the specific disease. So there are a lot of instruments that can wash these cells, take out all the chemicals that you added and clean them up, concentrate those cells and add into the right formulation. So you have instruments like Rotea that can, wash and concentrate cells. And then the electroporation system where you're electrocuting the cells and adding the genes. So we have instruments like xenon electroporation system that can take those payloads into the cell. Several cool technologies to deliver, to check, and make sure the right things are happening.
So what are some of the exciting gene therapies in the pipeline that are being developed that aren't approved yet?
There are several examples. As I mentioned, a lot of the early ones were this liquid tumors and oncology cancer treatments. What we are seeing is in the pipeline, there are diseases, several diseases lined up for cure, from anywhere from vision loss, muscular dystrophy, and autoimmune diseases as well. So several of those lined up in that preclinical to clinical trial stages. And it's looking forward to see all of those, get approved in one form or the other.
You're talking about very personalized treatments, and they can involve, as you said, extracting someone's cells, reprogramming these cells, and then sometimes making more copies of these cells, and then infusing them back into the body and doing all the quality control tests to ensure the edits have taken and the cells are now doing their job effectively. This does not sound like an off-the-shelf product. So how do you scale this to create a therapy that's available to treat thousands of people, hundreds of people, thousands of people, tens of thousands of people?
Great question. I want to introduce a couple more terms here, right? So one is personalized, right? So auto, self. So I take cells from my body if I have a problem, right? You take the cells out from the patient, engineer, make those gene edits, put them back. That's called autologous therapy. And the other example is where I can derive these cells, like the immune cells that I want to arm with new function to go hunt for the cancer and clear it. I can screen the healthy donor samples and then get those immune cells from donors. And then what I could do now is I can make these changes, right? The researchers or the clinicians, whoever are manufacturing these therapies, can make these changes in those healthy donor-derived immune cells and then expand them, if you want. Expand them to large enough scale. And then you do it once, you vial them into different dosages of whatever cell number per injection that you need, and now you have it off the shelf. So in these cases, you use specialized immune cells. You have to screen for those. So that's one way where you can move from autologous therapy, which can be expensive. And these therapies, when I say expensive, you're talking about anywhere from $500,000 to over $2 million per therapy.
Wow.
So if I can move that to allogenic, you can reduce the cost there. But there are some diseases where it's specific and niche for that particular patient where you might have to go back to the autologous therapies, but the field is moving towards that. And the other question that you asked, how do you ensure that is more, scalable across there? So the other thing is, you can optimize the processes from start to finish, right? And then ensure that these processes are tight enough so that it's reproducible. That's another benefit of going for allogenic kind of therapies. So where you have this process set, you manufacture it, and then you can lock it down versus making it one for one, you have one for many. And there are a lot of automation technologies that you could use to automate the process as well to drive down that cost and make it more scalable.
You mentioned automation. How is Thermo Fisher involved in automation?
You can break that into a couple things, right? So we are building instruments that enable automating, cell and gene therapies. So I mentioned earlier in our conversation about ex vivo therapy, where you take cells outside the body, you make those changes, you can grow them into whatever scale you want, whether it's autologous or allogenic therapy. you need instruments to do various steps. So we have instruments like our cell therapy grade, clinical research use grade instruments like Rotea, and then which actually does washing and concentrating of the cells prior to putting that back into the patient, and several steps ahead of that though. And then we have instruments for gene delivery. So those are the types of instruments, and I quoted some of those examples earlier. But we're also looking into, what can we do in enabling lab automation from end to end. So we're working with Multiply Labs. We have had some couple projects and papers where Multiply Lab is a robotic company that is bringing robotics to help automate the cell therapy manufacturing process. So we had done some collaborations with them. So that's one example. So, either building instruments or working with other robotic companies to enable those automation of cell and gene therapy manufacturing processes.
I understand that a lot of biotech companies, biopharma companies that are doing this, are particularly small biotech, right? This is the therapy they're developing is their expertise. So Thermo Fisher just is developing a network of collaboration centers to use its expertise and its equipment and technology to help with the process of scaling this up, correct?
Yes, we are. I mean, as I mentioned, we have a lot of tools that we provide, technologies. And then we have CDMO arms where, one could use our contract manufacturing teams to do that. Then we have the collaboration centers that you mentioned. And the nice thing about these collaboration centers is, as I mentioned earlier, these processes are intense, right? This starting from processing the cells to getting it to the body, making sure it works. So the main purpose of this collaboration centers was also to make sure that we work hand in hand with our customers who are clinical manufacturing companies, work hand in hand with them, provide our tools, and as we provide those tools, we also help optimize those protocols for our respective tools so that you reduce that barrier to entry with those protocols. So anything that you can do to help optimize those protocols for our tools will benefit the end user because that is so much less work for them upfront. Not to trivialize the extraordinary work that they have to do after taking the protocols, but that's what we do with these collaboration centers. And we have several of those, different parts of the world.
We talked about how this field has really grown leaps and bounds since the 60s, but it's still new. So what are some of the challenges that are still ahead of us that that you expect will be tackled to advance this kind of therapy?
There are several things, right? One, as I mentioned, you have to bring those costs down to make it more accessible to the broader patient population, not only those who can afford these therapies, and even, the prices are really astronomical. So automation is one of the key things. The field is moving there as well. So introducing robotic automation to integrate those processes, take out that manual processing. It does two things. One, a manual intervention adds more concerns for contaminations, right? So you take that out. Second, you are automating that process so that it's reproducible each time. And then the other thing is, you want to make sure there's a lot of diseases that are life-threatening and not easy to change. So there are a lot of improvements on the CRISPR gene editing technologies. Can this be introduced into the body directly? And then once it's put in there, how can it go efficiently and find the actual target where it needs to make the change? And the safety and efficacy of those tools. So a lot of research being done there. So I think the more these tools are optimized, the better it would be. A lot of exciting things happening in the delivery technology. I mentioned about viral vectors, and there are safe ways of, optimizing this virus so that it's not toxic. But then, moving away from those viral to non-viral based technologies like the lipid nanoparticles would be more exciting. So there's a lot of research being done there to see what kind of right tools I have. Can it deliver a broad variety of payloads? So quite a lot of things there. But in each of these cases, optimizing the tools, if I may say, so that it can work the first time right, moving it from manual to automated robotic-enabled processes so that, you reduce the cost and the manual labor and add that precision there. And then finally, really making it affordable,
Yeah.
moving beyond cancer to autoimmune diseases and other life-threatening diseases. It'd be really magical to see that evolve in that direction.
So you're the specialist in this field and you're very enmeshed in what's happening and what's being developed. But where is this headed? What's the future for this field that you're anticipating that we haven't even thought about yet?
I mean, as I said, moving into more disease types beyond cancer, because a lot of these approved therapies were oncology diseases. So going beyond that for more diseases, I would like to see more of that. And then improving those delivery technologies, improving those gene editing tools where those reagents don't become the barrier, those technologies don't become the barrier, the foundational technologies don't become the barrier, right? So I'm looking forward to those improved technologies that at one day, as I mentioned, a patient for a little child going to those hospital to multiple visits and that the really debilitating symptoms, if you can take that out with just one treatment, one dose and done, well, that's what I'm hoping for. And automation, of course, looking forward to how this can happen. And the other thing would be really decentralizing the manufacturing, right? Now it's being made in these niche facilities in one geographical location and then has to be transported and shipped somewhere. Just imagine a patient just waiting to get that therapy. And probably they have 2 weeks or 3 weeks. And this might be, a last line of therapy that they're trying. Mm-hmm. Can you reduce that time? So if you can decentralize manufacturing and get these therapies bedside, it needs a lot of, and that's something I'm looking forward to.
Well, that's a nice way to end this on hope and looking forward. And you really helped us understand the excitement about this field. Thank you for sharing your expertise and your insights today. And really, thank you for helping us understand this really exciting, complicated field.
Thank you, Terry. It was wonderful talking to you and discussing this exciting field.
Thanks for tuning in to this episode of Science with a Twist. This podcast is brought to you by Thermo Fisher Scientific. If you enjoyed this episode, then follow Science with a Twist wherever you get your favorite podcasts. Or join us online at thermofisher.com/sciencewithatwist for all of the latest episodes.